I am Dr. A.P. Gowsalya Devi, Assistant Professor of Botany, the Standard 5 Rajaratnam College for Women, Sivagasi. Today we are going to see about the Mendelian genetics, that is the basics of genetics, or what is the gene and all these things. Okay. Genetics. What do you mean by first genetics? Genetics is the branch of biology that deals with the heredity and variation of organisms. And heredity, it is the tendency of offspring to resemble their parents, is called heredity. So first you know, genetics is a branch of biology that deals with the heredity and variation of organism. Heredity, it is the tendency of offspring to resemble their parents. And we are not as a clone of our parents, isn't it? We just differ. We are not as such as our mother or as our father, and we, even though we are blood brothers and blood sisters, we are all having some differences, isn't it? That is what it is called the variation. That plays the role of the gene. That is the tendency of offspring to vary from their parents is called variation. Okay, that is why the field of genetics, it deals with the heredity and variation. That is, we are getting some characters from our parents, and we are also getting some variation from our parents. Here we should note one point, that is parents. They are not a single one. They are considered as a two persons, isn't it? So we are getting a combination of two characters from those two individuals. That is why there occurs the variation. We are not resembling as such our father, or we are not going to as such resemble our mother. Any organism, any living organism in this world, it will be like that only. So there plays the role of the gene, how it has been passed out from the parents to the offspring. So some of the characters we are getting from our parents, that is called heredity, and we are having some differences, that is called the variation. So what is the other things, we can see it now. Before that, the term genetics was first coined by William Patterson in 1905. Okay. And when you talk about genetics, we cannot omit the scientist Gregor John Mendel. We all know, isn't it? Yes, he's called as he's the father of genetics. Yes, just a small introduction about him. He is an Austrian monk born in Czech Republic in 1822 and went to the University of Vienna, where he studied botany, and he learned the scientific method. He loved to read, especially about natural sciences, and he is very much aware of Darwin's findings. And he studied the inheritance of traits in pea plants. There alone comes, of course we should know one thing when you think about Gregor John Mendel, at his period there is no any discovery of gene. He doesn't know what is a gene. He doesn't know how the characters are transmitted, but he gives a mathematical explanation. That is his more important things. Okay, prior to Mendel, heredity was regarded as a blending process, and the offspring were essentially a dilution of the different parental characteristics. Before Mendel's findings, what the scientific world believed, that it is a blending process, that is the offsprings has been gutted from the dilution of the different parental characters, but how the parental characters are getting, that no one knows. Okay, and he is the basic of genetics. Mendel, that is why he has been considered as the father of genetics. Okay, before going into the insight, we should know first. We first told, isn't it? It is the characters which has been transmitted from the parents to the offspring. How the characters are transmitted, how a newborn child has been born, how it is getting the chromosome, how it is getting the genes. Okay, for that we have to know about the chromosome, or in the condensed form it is called as the chromatids also. Where the chromosome or the chromatids are present, we should know about a cell. Each one of us made up of a huge number of cells, isn't it? Each one of us, any living organism, it is combination of cells. They forms the tissues, they forms an organ system, then form the organism. That what we are. So that each cell, I think everyone will know about a cell and cellular organelles. In the center of the cell, there is a particular part called the nucleus, and there, in the inside the nucleus alone, the chromatin network has been present. On the chromatin network, it will be condensed at the time of meiosis, that is during its replication. Mitosis and meiosis, cell division. So during the time of meiosis, it will undergo the condensed form called the chromosomes. There you can see one blue color and red colored things. They are called the homologous chromosomes. And there also you can see in blue color, you can see two tips. That is, in the centromere part it has been attached, and in the two sides it has been, you can see the splitted centromere chromatids. And in the red part also you can see. So it is called as the bivalent stage. Okay, that is during the meiosis. You might have studied this. So these chromosomes, they are the thing. Before that, of course, before Mendel and before the discovery of gene, of course in 1960, Watson, Crick alone, they discovered the genes. Before that, it has been believed that the chromosomes are simply carrying the characters from the parents to the offspring, because these chromosomes alone, only during the gametogenesis, they will divide and form two chromosomes, two gametes, and that gametes will be passed on to the formation of the zygote, that one gamete from the mother and another gamete from the father, they will combine together and form the zygote. And these chromosomes alone carry the hereditary information, that is the genes. How they are carrying and how the genes are responsible for the character. For that, in short, we can just know the gene is nothing but a segment of DNA. So where the DNA is there, it is in the chromosomes. In these chromosomes alone, it is the nucleoid chromosome model we may have studied. That is the histone proteins, it has been in like a cricket ball, and the DNA has been wrapped around like that, and the histone proteins. So from the DNA, the message has been passed on to the RNA, and the RNA to the proteins. You can just know it is the DNA structure. You can see the double helical structure. There, what is there? You may have see the letters A, T, C, G. They are called the nitrogenous bases: adenine, thymine, cytosine, guanine. And these nitrogenous bases, they are responsible for the transmission of the characters, very particular if you told, because these nitrogenous bases, they are responsible for coding the amino acids. These amino acids, because from the DNA, it will be through transcription, and mRNA will be formed. You can see here the single strand. Yes, from the DNA, one strand will be that will access a template, and an mRNA will be formed by the process of transcription. Then from the mRNA, by the process of tRNA, the translation process, and rRNA, that amino acids will be formed, and a lengthy chain of amino acids with the polypeptide bonds, it will be formed, that we called as the protein. So if any one amino acid is changed, the resultant protein will be changed. If when the amino acid will be changed, if one nitrogenous base is changed, then the resultant amino acid will be changed. As a result, the protein will be changed. So what will be what is the resultant end product, the protein, that a character will be expressed. That is whether it is the height of the plant, or color of the flower, or position of the flower, shape of the seed, whatever it is. Like that we might have seen very good example I can say. We can see in human beings, we might have notable differences. Like we are all of Asian origin. You can see the differences in Mongolians, Europeans, Negroes. So everything it is based on that alone. So it is the thing. The DNA, it acts as a hereditary material, and it is a central dogma. The DNA undergoes replication, it will form on, and the transcription, the mRNA will be formed, then by the translation the protein will be formed. In some material it will be the RNA will access the hereditary material, that is in retroviruses. There also the RNA will undergo the reverse transcript as DNA, then again to DNA to mRNA the protein will be formed. Okay. Chromosomes and genes occurs in pairs. They are called as homologous chromosome. What do you mean by homologous? Here you might know about, in human being we are having 22 pairs of chromosome, that they are body chromosomes, and the 23rd pair it is your sex chromosome. X and X it is in the female, and X and Y in the male. So all the 23 we have been numbered 1, 2, 3, 4 by the 22 pairs, isn't it? The first chromosome of the male and the first chromosome of the female, they are the homologous chromosome, like that. They will be of the same. How you can say that it is the same? The genes which are arranged on the chromosomes, which will be in the same order in the both the chromosome. That is the first gene. I already told gene is nothing but a segment of DNA. So a part of DNA which is responsible for the characters, which will be arranged in the same order. So that only during meiosis crossing over can occur, and the same set of genes will be transferred. Because if you in one chromosome it is in the part of an eye, in another chromosome it is in the part of the ear. If they get crossed, then it will be a new organism, isn't it? So that is why it is the homologous chromosome. It is all scientific proof. Okay, and here these chromosomes, you can see the dark blue, light blue, and all these things. So they are the pair of chromosomes, how they are arranged in both the thing. So these two chromosomes from the parents may undergo the fertilization and undergo the zygote formation. Just we know about the go into the thing, you just know about the terms. So gene, it is the unit of heredity, a section of DNA sequence encoding a single protein. That's what I explained so far. In the diagram itself, you can see the chromosome. From the chromosome, how the DNA has been, the DNA structure has been crossed, and the DNA, in the part of the DNA segment, it is responsible for a gene. That is, the gene will encode a protein. Okay, so that is what a unit of heredity abdina. Heredity na nammuடைய பாரம்பரியம் பலசில how we are getting the characters from our forefathers. Genome, the entire set of genes in an organism, it is called as the genome. Then what do you mean by alleles? I told, isn't it, the two chromosomes. If it is just take it in for an example, it is the A gene and A gene, the two genes that occupy the same position on the homologous chromosome that cover the same trait. That is, trait means a character. That may be height of the plant, shape of the leaf, or color of the flower, or whatever the thing. Those two genes are called as the alleles. And alleles, they are responsible. That is, a pair of genes are responsible for a character. Not a single gene can give a character. Then locus, what do you mean by locus? A fixed location on a strand of DNA where a gene of one of its alleles is located. Where this, I told, isn't it, it is the A, B, C, we can say, tell. That is the portion of the segment of DNA which is responsible for a character. Homozygous, that is having identical genes, one from each parent for a particular characteristics, they are homozygous. Having two different genes for a particular characteristics, they are heterozygous. What do you mean by this homozygous and heterozygous? If you are getting both, before that you can know about this term, that is the dominant and the recessive. The allele of a gene that masks or suppresses the expression of an alternate allele, the trait appears in the heterozygous condition. If it is a recessive, an allele that is masked by a dominant allele does not appear in the heterozygous condition, and it can appear only in the homozygous condition. That is, for each character, that will be there will be a dominant character and a recessive character. Just you can take for the height of the flower, height of the plant. If it is 10 meters, it may be a dominant character, and that is a tall plant. If it is a short of 5 meter height, then it is a dwarf plant. Here the tallness is the dominant character and dwarfness is the recessive character. When the tallness can be repressed, if in both, I told you, show you the allele, isn't it? Yes, in both the chromosome, if the capital T is present, we are just expressing the capital letter T. There is no any letter, it is for our assumption, we are giving the letter, okay. So if both the thing they are having the dominant thing, capital T, then the tall character will be expressed. If one of the alleles if one gene is having capital T and another gene is having small t, then also the capital T will be expressed. That is what the dominant. That is, it won't allow the recessive character dwarfness to be expressed. If both the genes are present as in the small t, then only the recessive character can be expressed. Okay, so that is what. So if both of them are having the capital T, then that is from both the parents. If we are receiving capital T, then it will be of homozygous. If from one parent it will be capital T and another parent it will be of small t, then it will be in the heterozygous condition. Okay. What is the genotype? The genetic makeup of an organism. The phenotype, it is the physical appearance. Phenotype, what we can see with our naked eye. Genotype, what is the genetic, whether it is in the homozygous condition or heterozygous condition. That is alone the genotype. Okay, that is the genotype plus environment phenotype. Okay, what Mendel did, he studied the traits that occur in distinct forms. He developed true breeding varieties. When bred among themselves, these plants produced offspring of identical of the parent for the trait. And the most important concept here, he used mathematical analysis in his studies. What he did his studies, we can see now. Now we can see the height of the flower, the color of the flower, height of the plant, the pod shape, seed coat and all these things. Like that he had taken in pea plant, because in the pea plant, he undergo all this pollination. How he did his experiments in pea plants mean, I told you, isn't it, he did by his mathematical experiment, used mathematical analysis in studies. How? In the first year, he sown some 100 seeds. All the seeds which have been taken from the tall plant, that is called as the parental generation. And in the when he sowed and the plants grown, and it is called the first generation or F1 generation or filial generation we used to call. There he crossed the tall plant and the dwarf plant. How he crossed, okay, that we can see now. So before that we should know about the simple terms here. So normally how the plants undergo fertilization and the formation of zygote. So it is the gametes, the reproductive cells produced by sexually reproducing organism. In plants, it is a flower of a pea plant. You can see the calyx, corolla, and the main part it is the androecium and the gynoecium. There you can see the pollen grains and the stigma, the ovary with the ovules. Okay, the male gametes, it is the sperm in plants contained in the pollen grains. In female gametes, it is the eggs in plants contained in the ovules. The ovules contained in the carpels. Okay, fertilization, fusion of egg and the sperm. Self-fertilized, fusion of sperm and egg from the same plant. If it undergo fertilization, it is a self-pollination. If fusion of egg and sperm from two different plants of the same variety, if it is a pea plant alone, then it is a cross-pollination. They produce the hybrids. The first one, it is the F1 generation. The second one is the F2 generation. How he did it? So it is the experiment. He did it by this. He take the two plants. He did it by emasculation experiment. That is, the first plant he considered it as the male plant, and the second plant he considered as the female plant. So in the female plant, whichever he considered as a female plant, he removed that is called the technique called emasculation, removal of anthers. He removed it and make the pollen grains of the first plant to be dusted on the female part of the second flower. So now the second plant cannot undergo self-pollination because the male anthers has been removed from the second plant, and the anthers of the first plant has been dusted. He considered the first plant as the tall plant and the second plant as the dwarf plant. Now he made the cross pollination by crossing the tall and the dwarf variety, and now the plant grown and it will give the seeds. Now these seeds he just go it for this thing. He undergo the seeds to undergo self pollination alone. He didn't do the cross pollination. When these F1 seeds, tall and dwarf, he crossed, and in the F1 seeds he got all the plants as tall plant alone. He just get embraced. Where goes the dwarf character? Then when it undergoes self pollination, these F1 plants, there he comes, he got the tall and dwarf plants. Okay, so then he find out the results as he just gave only the results like that alone. That is, I sown 100 plants by the emasculation methods. In the F1 generation, all the 100 plants grown were tall, but in F2 generation, I got tall and dwarf in the ratio of 3:1. That is 75:25. That mathematical explanation alone he has given. Okay, he was the first biologist to use mathematics to explain his results quantitatively. The science in the science mathematical experiment, through the mathematical experiment, a science concept has been explained only by Mendel. He predicted the concept of genes, that the genes occur in pairs, that one gene of each pair is present in the gametes, but he doesn't know anything about gene. So he just gave in his concepts, it is some factor, it has been responsible for the transmission of character. Okay, that is the hereditary information that determines a single trait. Alleles, it is an alternative form of a gene. When an organism inherits two identical alleles for a trait, organism is said to be homozygous. What I explained? For two different alleles for one trait, then it is called as the heterozygous trait. In 1866, he published his experiments in plant hybridization, in which he established his 3 principles of inheritance in the annual proceedings of the Natural History Society of Brunn. Of course, his work was largely ignored for 34 years because he has published in 1866 only in 1900 3 independent botanists they rediscovered Mendel's work. After his death, we should know. By the 3 scientists, Karl Karens of Germany, Hugo de Vries of Holland, who is called as the father of mutation, Erich von Teschermark. So Mendelism or Mendelian has been found out by them. Thank you for watching this video. In the next video, we can continue this.